Thank you. Once again, very good evening to all. Yeah. Today I have to talk about the topic on Vibrio hobby, genomic insights into a NGS approach. So, in this specific topic, I would like to talk about the following contents. So, first of all, the basic about the shrimp aquaculture scenario at present and what are the challenges we are facing in shrimp aquaculture. Then the main focus on Vibrio bacteria. In this Vibrio bacteria, I have to talk about more specifically on Vibrio hobby. Then what are the NGS platform used nowadays? And coming to the topic, I would take the whole genome sequencing for Vibrio hobby. So in this specified topic, I would like to talk on various gene production method and what are the gene functional annotation methods I have to characterize. Characterize in the specified Vibrio hobby. So all the details should be talk about in this specified topic. Okay. Then finally, result and discussions. Okay. So coming to the shrimp aquaculture scenario. So very basically, according to the Food and Agriculture Organization in 2018, aquaculture has emerged as one of the world's most promising sector towards seafood production. Most importantly, shrimp aquaculture sector deals with broad spectrum activities related to food production and nutrition. So, in worldwide aquaculture sector, the white-leg shrimp Lithophilus vanami is the most important crustacean species, particularly for the developing coastal countries. So, according to International Market Analysis Research and Consulting Group, IMARC, reported an Indian shrimp market reached a volume of 0.74 million tons in 2019. Basically, shrimp contains high nutrient value. It's including protein, selenium, antioxidants, phosphorus, magnesium, copper, iron, and vitamin B12. So, in Indian scenario, by species level, Venus vanami, Lithophilus vanami, and Venus monodon is the dominant species cultivating in our country. So, more specifically in the region of West Bengal, Andhra Pradesh, Tamil Nadu, and Pondicherry, Orissa and Maharashtra, and Gujarat is the most important regions where cultivating this kind of species. Then, the leading newspaper, Times of India, survey in January 2021 reported nearly 70,000 farmers are cultivating Lithophilus vanami in India and almost produced 7.11 lakh tons in Lithophilus vanami production in 2019 and 2020. Also, 74% share of shrimp in overall seafood productions. And almost 46 crore value of seafood exports in 2019. So coming to the challenges in shrimp aquaculture has faced so much problem. So very basically for the first one is the cultured shrimp suffer various diseases due to infectious and non-infectious diseases. So coming to the infectious diseases caused by viruses, bacteria, fungi, and parasites, and non-infectious diseases caused by adverse environmental conditions and nutritional imbalances and some genetic disorders. So besides the enormous environmental factors in shrimp farming, including inappropriate environmental conditions such as poor soil and water quality, high density stocking with very limited water exchange, nutritional imbalances, inadequate aeration, and mismanagement of daily operations are depreciating the yield of shrimp aquaculture sector. So the diseases are leading the mortality rates about 80% of making huge losses to the shrimp aquaculture sector. So in case of shrimp aquaculture, many diseases are spread rapidly throughout the year. So in case of majorly for viruses causing white spot syndrome virus, Then bacterial diseases, in case of white gut syndrome, white feces syndrome, then microbial enterotoxin, mycotoxins, and majorly vibriosis is the main diseases caused by the aquaculture industries. So there are several disturbance of pathogen host environmental interaction progress to disease in aquaculture. So in shrimp aquaculture, majorly the 6 factors were responsible for causing diseases. There are pathogen, host, nutrition, physiological factors, environment, and husbandry. So here, the outbreaks in association with Vibrio species in shrimp aquaculture, they majorly the following diseases caused by the Vibrio species. In case of eye lesions and detachment of midgut epithelium, Boletus nigricans, white gut disease, bright red syndrome, white tail disease, red leg disease, invasion of eggs and larvae, and lucial disease, red disease, and finally luminescent vibriosis. All these diseases caused by the Vibrio species. Most predominantly, Vibrio harveyi is the one of the bacteria causing this all kind of diseases. So coming to the talk about the Vibrio, first of all, what are the Vibrio bacteria? So coming to the taxonomic classification, the genus is Vibrio, the family comes under Vibrionaceae. In the order of Vibrionalis, the class comes under Gamma Proteobacteria, and the phylum is Proteobacteria. Generally, Vibrio is a diverse genus of bacteria that exists free living or associated with human and animal hosts. Generally, Vibrios are gram-negative, motile rods, ubiquitous marine bacteria. The size is around 1.4 to 2.6 micrometer in length, and it's generally facultative anaerobic bacteria. So Vibrios is a phenomenal test model for the genomic classification because they are abundant in the aquatic environment and it's naturally occurring in marine and coastal waters. So it's grown mainly for halophilic condition. So grow within very high salt concentration, and it's also present in high salt concentration during summer, then high growth rates in warm temperature. So this is the bacteria, the fastest growth rate of all known bacteria. So in year of 2020, Tanaka et al. Reported more than 130 recognized Vibrio species. Generally, Vibrios are associated with shrimp aquaculture practices, often causing significant economic losses. Here, Vibrio harveyi reported as recurrent pathogen in shrimp hatcheries. Then the Vibrio harveyi frequently infect the shrimp, causing very large number of diseases. Vibrio infection could go. Vibriosis is the most widespread disease infecting various aquatic organisms, including fishes, mollusks, and crustaceans worldwide. And the pathogenicity, the virulence factors of Vibrio harveyi are not completely disappeared. In case of extracellular products, including proteases, phospholipids, siderophores, and hemolysins or exotoxins are considered to cause pathogenicity. In recent years, the development of next-generation sequencing has enabled multiple genome sequencing and assembly of Vibrio species. More recently, there are several bioinformatics and computational tools are successfully applied to obtain the insights into genome architecture of specific organism, genome environment interaction, and establishment of evolutionary models. So coming to talk about the Vibrio harveyi, is generally a bioluminescent bacterium and gram-negative curved rod-shaped marine bacterium found in the marine communities, and it is a predominant pathogen that induces 100% mortality in the course of early larval stages of shrimps, resulting in enormous economic losses in the production and marketing of aquaculture shrimp. And also, Vibrio harveyi commonly contaminates various species of fish, crustacean, mollusks, and other seafoods. So the genomic characterization of Vibrio remains quite challenging as a consequence of selection forces, dependent genomic plasticity, and lateral gene transfer manipulation in the form of environmental adaptation. So now we will talk about the NGS. First of all, we would know about the whole genome sequencing. So here, what are the main importance for whole genome sequencing here means, first of all, we should know about the coding and non-coding part of the genome. Then we can retrieve the important pathways, and also we should know about the evolutionary study and species comparison, then identification of important secondary metabolite pathways, then finally disease sensitivity production based on gene sequence variations. So these are all the important factors to know about the by using whole genome sequencing. So in the right side figure, I have illustrated some milestone and significant milestone in the DNA sequencing. So in the year of 1869, a discovery of nucleic acid by Friedrich Miescher, followed by in the year of 1953, discovery of the structure of DNA double helix by James Watson and Francis Crick. In the followed by the year of 1965, Robert Holley determined the first alanine transfer RNA nucleotide sequence from yeast. Then the year of 1976, the first complete DNA genome was sequenced in bacteriophage. Followed by in the year of 1977, Sankar and colleagues described DNA sequencing by enzymatic synthesis, and Maxim and Gilbert demonstrated DNA sequencing by chemical degradation. In the year of 1980, Nucleotide Sequence Library, European Molecular Biology Laboratory, EMBL, was established. So followed by in the year of 1982, GenBank started the public database for DNA sequences. Then the year of 1983, we all are known about the polymerase chain reaction was invented by Carey Mullis. Then the year of 1986, the first semi-automated DNA method was developed by Laurie and Smith at California Institute of Technology. Then the year of 1987, Applied Biosystem developed a fully automated DNA sequencing machine that is called ABI 370. Then the year of 1990, Human Genome Project was initiated. Then the year of 1996, Applied Biosystem developed capillary DNA sequencing platform. Then the year of 1998, the model organism C. elegans genome was sequenced. Then followed by in the year of 2000, a Drosophila genome was sequenced. Then in the year of 2006, Illumina sequence was introduced. So after Illumina sequencing was introduced, there are several publications or outcome in the field of next generation sequencing. So in the year of 2011, BacBio SMRT sequencer was introduced. Then finally, in the year of 2050, the Axport Nano4 Minion sequence was introduced. So this is all the very important, significant milestone in the DNA sequencing. So coming to the whole genome sequencing process, so these are all the main important steps involved in the DNA sequencing technologies. The whole genome sequencing technologies. The first of all, we have to take the bacterial culture, then go for the DNA extraction. The third step is DNA sharing. Then fourth one is we have to construct the DNA library preparation. Then the constructed DNA library was go to the sequencing machine. Then finally we can get the DNA sequence. So we'll get the DNA sequence by using some bioinformatics tools. We have to annotate the results. So these are all the very important steps involved in the whole genome sequencing. So coming to the whole genome sequencing of model organism, we assume that so first we have to go for the any organism or any plant source or whatever, any model organism, we should know about what is the genome size. So that is very important. So once we will know about the genome size, then only we can go for the how much data we have to generate it. So then only we can finalize it. So that's why I have illustrated this figure on already sequenced model organism. In case of prokaryotes, E. coli, so the genome size is approximately 2.6 megabase per. The number of protein coding genes is almost 4,300. So in case of eukaryotes, is the genome size is in case of Saccharomyces cerevisiae, the genome size is almost 12 MPs, and the number of protein coding genes is 5,800. Then C. elegans is almost genome size is 100 MP. The protein coding genes is approximately 20,000. Then Arabidopsis thylana is 125 MPs. The protein coding genes is 27,000 protein coding genes, that is CDS. Then Drosophila melanogaster is genome size is 120 MP. Then the protein coding genes is almost 13,000. So this is the very basic idea about the genome size and how much protein coding genes involved in these particular organisms. So what are the comparison between Sankar sequencing and NGS technologies means? So we should know about the Sankar sequencing is a very traditional method. It should be used for several NGS applications in the previous years. So after that, the next generation sequencing has come into the mind. There are several sequencing technology has improved. In case of comparison between these two, first of all, the sample input for the Sankar sequencing is purified PCR product or plasmid used. Then in case of next generation sequencing, NGS library constructed from genomic DNA or amplicons was used. Then the sequencing chemistry for Sankar sequencing is electrophoresis. So coming to the NGS technologies, the sequencing chemistry is for to determine the sequence of each DNA molecule during the synthesis. Then data capture for Sankar sequencing is only homogeneous DNA sequence was identified. In case of next generation sequencing, there are several numerous DNA sequence determined simultaneously. So what is the main advantage for Sankar sequencing is very high precision, simple data analysis. A process will take very long weeks. Okay, so that is the very important advantage for Sankar sequencing. So coming to the next generation sequencing, it's very efficient for different massive sequence determination by the single run. So coming to the disadvantage for Sankar sequencing is very difficult to analyze heterogeneous amplified products in sample and very slow process. So what are the main disadvantage for NGS is the advanced bioinformatics analysis was used and data interpretation and analysis is somewhat difficult. So what are the different NGS platforms are used in the nowadays means there are several sequencing platform was used. The major sequencing platform for Illumina is Minisec, MySec, NestSec, HiSec and NovaSec. The sequencing chemistry for Illumina is reversible titanometer. Then the library preparation is basically for bridge amplification. The read length used for Illumina sequencing is basically for 100 to 150 base pairs. This very short reach. Then it's used for basically for HiSec sequencer. Then 400 to 500 for MySec sequencer. The sequencing time for Illumina is 1 to 6 days. The output data for run is 500 to 1000 GPs. Then what is the main advantage for Illumina sequencing is very high precision and low cost per base and least error rate. So compared to other platform, the error rate is very, very low. So that way we all are prepared for the Illumina sequencing. Once we will go for the nanopore sequencing, when we go for any we can go for the long read sequences. If we can generate more number of data in the sense, we should prepare the nanopore sequences. So these are all the main important sequencing platforms. The Illumina, the iron tolerant and BacBio and nanopore is the sequencing technologies used in the NGS platforms. So coming to the overview representation of a study, here I have collected the shrimp from aquaculture pond located at Morakonam, east coast of India. Then go for the serial dilution. Then after serial dilution, we can go for subculturing. So I can finally get the pure culture. Then pure culture we can inoculate into the albibroth medium. Then we'll go for the DNA extraction. So after DNA extraction, we have constructed the DNA library construction. So in this DNA library construction, so DNA is first of all fragmented. Then we can form the DNA library construction. So after DNA library constructions, the library is inserted into the sequencer. So the sequencer has generated 1.3 GP of raw reads. So after that, we are getting the raw reads. After the raw reads, we can filter using trigonometric and adopted cutter tool. So after that, we can get the filtered data. That is called the quality controlled data. So after quality control data, I have to assemble the data by using space and velvet optimizer. So that is called a de novo sequence assembly. So after assemble the data, I can protect the gene protection. So in this gene protection, I have protected coding genes. What are the protein coding genes present in this genome? So after that, rRNA and tRNA. Followed by gene functional annotation, also protected by using a cluster of orthologous, gene ontology, and Kyoto Encyclopedia of Genes and Genome, virulence factor database, inserting sequences, and POG regions. So all this genomic characterizations I have done in the Vibrio genome. So in case of a DNA library, I have used a TrueSec nano-DNA library preparation kit. Then the quality of final amplified library was validated by Agilent bioanalyzer. The sequencer was done using Illumina HiSec 2500 platform. So this is the DNA library construction. This is the steps involved in the DNA library construction. First, we can take the genomic DNA. So after that, we should go for the fragmentation. Then we can get the below 800 base pairs of DNA fragments. Then the DNA fragments are blended. We can get the blended fragments. So after the blended fragments, we can add the adenine at 3' frame. Then the 3' overhang, we can add. They can add and ligate the adapters. They can get the fragment with adapter at both the ends. Then finally, removal of unligated adapters. Then we can finally get the purified fragments with adapter at both the ends. Then we can go for the PCR amplification. Then finally, we can get the genomic DNA library construction. So after that, we can get the DNA library. We can go for the sequencer. So after sequencer, we can get the raw data. So after raw data, we can go for filter the data by using several bioinformatics softwares. Followed by the another next slide, we can discuss about it. Okay. So here, these are all the online tools. There's all the online database and tools used in this study. So first of all, Proca was used for gene annotation. Then Protocol, the prokaryotic dynamic programming gene finding algorithm used for protein coding sequences. This is the Protocol is used to find the protein coding sequences in the genome of Vibrio. Then the RNA was used to find the rRNA genes. Then Oregon was used to detect the tRNA and the tmRNA genes. Then COG database was characterized by orthologous relationship of gene consequences. Then gene ontology database was protected by GC protector, which will be represented for gene and gene product traits across all species. Then KEGG database was mainly used for pathway identification. Then PROPHAGE region was identified by using FAST search tool, that is FAST. Then to find the inserting sequences by using IS-Saga tool. To find out the virulence factors genes involved in this Vibrio genome by using virulence factor database. Then finally, the phylogenetic tree was constructed by using MIGS software. So this is all the software and online database tool used in my studies. So this is all the data, very minimum information about a genome sequence specification of Vibrio. So I have submitted my sequence archive in the database, in the NCBI database, and followed by the NCBI ID SRA number SRR 5410471. This is my bioproject ID and biosample ID. You can refer my if you want anybody to refer my sequence in the NCBI database, you can go for the SRA and give the accession number to get the raw data sequences. Okay. So my project name is Vibrio Auto6 genome sequencing and assembly. The geographic location for sample location is already I told, right? East coast of India, Maracana. Then isolation source is aquaculture pond. And also the project relevance is environmental and biotechnologicals. Then I have selected the read type of pattern sequencing. The read length is almost 101 base pairs. The fold coverage is 115x. What are the assembly method I have used means S-place and velvet optimizer. Then what are the source of file I have get means GFF35 and NCBA complement GenBank and FASTA sequences. So this is all this information available in the NCBI database. So coming to the one second. Yeah, coming to the genomic statistics of Vibrio hobby, the first of all, the quality assessment tool was used. The genomic feature of Vibrio hobby is generally the estimated genome size is 6.3 megabases and the GC content is almost 47.4%. The number of protein coding genes present in the Vibrio hobby genome is almost 5,730. The total predicted gene is 5,912. Then tRNA is 114. Then rRNA is almost 11, and miscellaneous RNA is 57. Here, GC helps to determine the thermostability. Its range between 38 to 72. It's indicated isolate diversity. Here, rRNA can vary among different species. It's generally between 6 to 19 genes are involved. So this is the overall genomic statistics of Vibrio hobby genome. So coming to the orthologous of gene annotation of Vibrio hobby, COG annotation. Basically, COG is an effective tool for the annotation of functional proteins. Here, COG categorization was found out by using HMM profile of gamma proteobacteria found in the popular egg NOG database. That is evolutionary genealogy of genes, non-supervised orthologous groups. In this Vibrio hobby genome, I have get the 23 classes of COG classification. Then finally I can get the 4,963 unigenes annotated on COG functional groups. So here the category is divided into 4 main categories, followed for the name of information storage and processing, cellular process and signaling, metabolism, and poorly characterized. So according to the metabolism, majorly 37.51% of genes are involved. Here, in the functional classification, inorganic ion transport and metabolism is the predominant genes. Almost 625 annotated unigenes were involved. So followed by the cellular process and signaling categorization, almost 25.23% unigenes were involved. It's in the case of cell wall membrane and envelope biogenesis involved 530 unigenes. Then followed by transcription, 516 annotated unigenes were involved in the information storage and processing. So this is a figure indicated majorly 37.5% indicated metabolism and 25.23% indicated cellular process and signaling, and 12.56% represented information storage and processing, and 24.68% poorly characterized unigenes. So coming to the gene ontology, so gene ontology is an important factor to international factors to find out the gene function of gene functions. So here gene ontology is comprised the 3 important categories. The first one is biological process, then cellular component, and molecular functions. So in this genome of Vibrio hobby, the annotated genome is almost 1,088 unigenes were annotated. In case of biological process, only 19 unigenes were annotated. In the case of a cellular component, only 15 unigenes were annotated. Then the molecular function, 9 unigenes were annotated. Both the 3 combination, biological process, cellular component, molecular function, almost 961 unigenes were annotated in all the 3 categories. So this is the predominant gene ontology terms represented in each of the 3 major GEO domains. The first one is biological process. Here, cellular process is the most predominant. It's almost 2,607 unigenes were annotated, and also the least one is a biological addition. Okay, in case of biological process. So coming to the cellular component. Here, cell is the most predominant unigenes. Almost 825 unigenes were annotated. The least one is extracellular matrix spot. Almost 4 unigenes were annotated in this spot. So coming to the biological molecular function, here catalytic activity is the most dominant category of unigenes involved in this category. Almost 616 unigenes were involved. The least one is enzyme regulator activity. Here 7 unigenes were involved. So coming to the Tato encyclopedia of genes and genome annotation pathway is an important biological function of genes through the identification of enzymes and other proteins in the biological process. And also it's a KEGG is a very metabolic pathway annotations. So to protect the genes and also identify the protein sequences of genes through KEGG automatic annotation server. So in case of KEGG pathways, almost 3,401 unigenes annotated. In the case of Vibrio genome, it will be categorized into 210 pathways. So these are the top 20 KEGG pathways associated with the Vibrio genome represented in this figure. So here, the most predominant one is metabolic pathways. Almost 557 unigenes were annotated in the case of metabolic pathway in the KEGG pathways. So followed by biosynthesis of secondary metabolites is almost 249. Then microbial metabolism in diverse environment is 165. These are the most predominant KEGG pathways involved in case of unigenes. The least one is glycine. It's almost 31 unigenes were annotated in case of KEGG pathways. So coming to the pathogenesis and virulent factors of Vibrio genome. So the genome of Vibrio encodes the various types of virulent factors, including the manner-sensitive hemagglutinin, MSHA, type 6 phyla genes, flagella-based motility genes, LVXX gene-based quorum sensing, pyoviridin, capsular polysaccharide genes, fibronectin binding proteins, EPS type 2 and virulence-associated secretions, that is VAS gene, and type 6 secretions, VGR2 and VASE. So this all comes under the virulence factors of Vibrio genome. By using the VFDB genome virulence factor database, in the case of Vibrio almost 36 virulence factors were determined. So here, so this was the virulence factor genes involved in the Vibrio genome illustrated in the table. So here almost 36 virulent genes were illustrated here. So this is the circular plot of the genome of Vibrio illustrated by using blast string image generator, the version of 0.95. The outermost layer is the GC cloud and innermost layer is the IS elements. Okay, so here 23 genes were responsible for the flagella-based motility functions. It's encode 8 FLI genes. The 8 FLI genes are namely FLIO, FLIG, FLIG, FLIP, FLII, FLIF, FLIQ, and FLIR. So these are the 8 FLI genes involved in this Vibrio genome. Then followed by 3 FLG genes, that is called FLGN, FLGM, and FLGI. The followed by 2 FLE genes, that is FLER or FLRC and FLEQ. Followed by 2 FLH genes, that is FLHA and FLHB, and 2 MOT genes, motility, that is called motility genes, MOTB and MOTY. Then 2 secretion genes, that is 2 SCR genes, there is SCRA and SCRC. Then 2 LAF genes, LAFS and LAFU or MOTB. 1 FLRA gene and 1 FLAE genes. So here, moreover, 3 virulence factors were subjected to manner-sensitive hemagglutinin, that is MSHA. It encodes MSHK, MSHM, and MSHF. And also 2 type 4 phyla genes. It encodes PILC and PILQ. Then finally RMLA and WBFV or WCVB genes encodes the capsular polysaccharide functions. So this was the 36 virulence factor genes involved in the genome of Vibrio. So coming to talk about the prophage characterization. The prophage was identified by using FAST tool, that is PHAST, FAST. So here one incomplete phage regions was identified in the genome of Vibrio. The total length is around 24.2 kp in size. the proposed types identified based on the score. Here, incomplete region is almost below 70 is indicated as incomplete region. The questionable is 70 to 90. Then the score is 90 is indicated the more than 90 is indicated the intact regions. So here, these are the pause elements annotated based on the regions. Here, by using lysis, terminase, and partial protein, protease, coat protein, tail shaft, attachment sites, integrase, pause-like protein, hypothetical proteins, and other and transposas, fiber protein, plate protein, and tRNA. So these are all the annotation pause elements identified in the prophage regions. Here, 15 number of hypothetical protein was identified, and also 24.2 kp in size in the genome of prophage regions identified. So here, this is the annotated pause elements identified in the incomplete region of Vibrio harvey genome. So coming to the IS elements, here inserting sequence was identified by using IS Saga tools. So IS Saga provides a high quality semi-automatic annotation system and accurate identification and annotation of inserting sequences. Here, inserting sequences are genomic regions within there are travel within or across the chromosomes. Generally, IS are intracellular mobile genetic elements which play an important role in the host genome evolution through a series of gene disruption and inactivation, resulting in a genome reductions. Okay, so here totally 30 number of inserting sequences were identified in the genome of Vibrio harvey. So finally, summary and conclusion of my study is, so generally Vibrio harvey is a well-known aquatic pathogen causing vibriosis infection in the shrimp aquaculture. The whole genome sequencing and bioinformatics analysis of the predominant disease-causing organism in shrimp aquaculture was sequenced, assembled, and annotated. Furthermore, this information revealed a high-quality data and genome assembly, which provide a more accurate gene annotation, genomic characterization, phylogenetic position, and taxonomic classification of Vibrio harvey strains. Then this genomic information was provided the significant molecular insights for ameliorating the understanding of Vibrio harvey genome. Then more specifically on the subject of evolution, defining functional classification such as cluster of orthologous, gene ontology, then Kyoto encyclopedia of genes and genome, prophage regions, inserting sequences, and also in pinpointing features that contribute to virulence factors in the pathogenesis of vibriosis. So finally, this finding of the study revealed that the whole genomic analysis approach provides reliable information on molecular and biological pathways influencing pathogenicity for developing new diagnostic approaches and therapeutic agents for effective prevention and management of vibriosis infection in shrimps. So this is my publication published in the Journal of Molecular Phylogenetics and Evolution. So under the impact factor of 4.4 in the year of 2020. Thank you. Thank you for listening to everyone.
Thank you, sir. Participants, if you have any queries, please post in the chat box or just raise your raise your hand option. I will unmute you.
Participants, do you have any doubts? You can feel free to ask. I'm ready to answer your questions.
Participant, if there is any question, please post in the chat box.
I think there is no questions from the audience, it seems.
Yes.
Anyway, thanks a lot for the presentation, Dr. Thirugnanasambandam. Actually, I have only one question.
Yeah,
so out of this, you have find out some virulent gene, but
yeah.
whether it will be very in what way it will be helpful to the aquaculture industry?
Yeah, yes, it's a very good question. Actually, here in case of Vibrio harvey genome, I have identified 36 virulence factors genes. Once we will go for the large number of bacteria, for example, we will go for basically, and Vibrio vulnificus is causing wound infection for farmers. Then the several bacterium like Vibrio paramyleticus and Vibrio vulnificus, then several databases. First of all, we can calculate and accumulate all the virulence factors genes. Then we can find out the what is the major pathogenicity causing virulence factor genes. Then we can accordingly we can go for the therapeutic approach. So the therapeutic.
So
and,
you mean to say that
yeah.
it's a virulence-based therapeutic approach?
Yes, sir. Exactly so.
Okay. Thank you. Thank you.
Thank you, sir.
Okay, if there is any no questions from the audience, we can go to the next session. Actually,
Good
the
evening,
next.
sir. I
Yeah.
have a question.
Yeah,
So
please.
since you said that we will come to know about the virulence factor alone, but how do you think it is applicable when it comes to the real hands-on aquaculture practice, especially with the shrimp? Because there the main problem is with the Vibrios, no? So is there any possibility that we can develop something where we can control the Vibrio count, or any sustainable measures we can take based on this NGS?
Yeah, exactly. Is a very good question, ma'am. So first of all, we should know about the for which bacteria causing what kind of virulence factors involved in this kind of bacteria. First, we should know about in our mind. Then simultaneously, there are some possibilities for a therapeutic approach. By using NGS technologies, we can go for it, ma'am.
But sir, already it is known that Vibrios have been, there lots of publications have been there which is mentioning about Vibrio being harmful. I mean the strains or the specific species being harmful to the aquaculture practices, especially for the shrimp and all. So
Okay.
in that case, when it is already known, how much it is possible that into the field we can useful for the farmer, because it's the main generation, right? So on perspective, how much or what they can do, because even if when you're saying therapeutic aids to
Voice is breaking. Voice is breaking, ma'am. Voice is breaking. Ma'am, voice is breaking.
Sir,
Yeah.
sir, can you hear me now?
Yes, ma'am.
Sir, can you hear me now?
Yeah, yes, ma'am. Yes, ma'am. Please
Sir,
tell me,
see,
ma'am.
you have mentioned about the therapeutic agent, but from the lab or from the analysis reaching up to the therapeutic agents after the trial version and everything reaching the farmer in hand as a therapeutic agent, it is still a lot of time span, everything is required. And we have a lot of informations about the specific strains which are already causing these issues for the shrimp farming.
Okay.
So in that case, already these diseases are already existing, or any equilibrium is being disrupted, it is going to create a issue or a disease for the shrimp.
Okay.
So in that case, how much it is possible that it can support the farmer, because farmer is more interested in getting lot of sustainable, more than sustenance, it is they are only looking into the production or the crop maximum production. So in that case, how they can think about these factors being suppressed, or anything we can do for
Actually, ma'am, first of all, we should go for the NGS technologies for in case of vibriosis or vibrio causing diseases. First of all, we should know about the genomic adaptation of the bacteria. Okay, we should know about the genomic adaptation. Then finally, we can go about the pathogenicity of virulence factor genes. Okay, so once the pathogenicity, we can identify the virulence factor genes. So what are the types of virulence factor genes? For example, in case of quorum sensing and pyrovidin and pseudophore genes, then we can go for the therapeutic approach. Then the therapeutic approach, we should go about the several anti-virulence therapies. Like there are several anti-virulence therapies available. So accordingly, we can applicable to the farmers.
So actually, adding to this, 1 minute, Actually, adding to this answer of Dr. Thiru, I just want to give some of the findings. What we have combinedly done some work. Actually, I just want to narrate here. Actually, all the Vibrio harvey, for example, all the Vibrio harvey, whatever we have isolated from the farm or whatever we are having in the lab are not pathogenic. The same strain, if we are challenging this particular, for example, it is a VH1 means, if we are challenging the VH1 with some kind of shrimps, it won't do any harm to that particular host. But the same strain we have isolated as pathogen from the pond, but when we are doing it in the lab, it won't work. So that's what we have gone with this particular genomic based work. Yeah. The biggest thing is Vibrio harvey as well as other Vibrio pathogenic strains. They work depends upon the environment. The virulence factor will be expressed depends upon the environment of the pond environment. So first of all, this kind of database will help us to know what is the percentage of such kind of virulence factor is being doing the predominant role in the pond. That's what finally I asked one question to Thirugnanasambandam, why we have done the work. He said that it is anti-virulence virulence-based therapy. Yeah. This is the technique what we want to done in our lab, and this is the thing what we want to give to the farmers. But it is not an easy joke, as you said. It is not going to work as it is in a single table or a single lab experiment. It needs a collective data, but every baseline data will be input for the final antivirulence therapy if we are making a new kind of therapy to the farmer. That is the answer.
Yes. Thank you, sir.
Thank you, sir. Actually, I specifically wanted to know, like, what will be the equilibrium for the environmental variables to act as, to support it, or otherwise where it can go from the optimal level, or where it can trigger the pathogenicity for the Vibrio harvey or any other species of Vibrios to act a disease-causing environment in the pond, like. That is why I asked this question.
Almost all your answers are based on the time. You are asking about any particular time in which the equilibrium will work. But as we are all working in biological samples and biological environment, it all depends upon the biological factor as well as the abiotic factor, which is correlating with the host. Yeah. So we cannot say the exact equilibrium for the same species which we are working. Though Thiru is having a strain with him, maybe it may change the nature of its virulence when it is exposed to a natural environment, pond-based environment also. So it is not possible to correlate the equilibria, what you said. It will vary accordingly to the environment. But we can come to a conclusion, what is the percentage of the particular virulence factor? The virulence factor, expression of the virulence factor, that definitely have a equilibria. That we have to find out. But with a single gene or with a single set of virulence factor work, we cannot done that. It need a collective or a global based virulence factor genomic database that will give the answer for your equilibria hypothesis.
Okay. Thank you, sir. I just asked because I work in this area. So that is why I thought like I will ask this question. Especially I work with the environmental parameters of shrimp aquaculture. And it's bioremediation. So that's how I happen to ask this question. Anyway, thank you so much, sir, for the clarification.
Anyway, for you and for the other audience also, if anybody want to do any kind of value addition with the genomic database set we are having with us, as now you asked, is it possible to work on any kind of other environmental parameter to work with a particular gene? If anybody is having a bioinformatics based value addition job to be carried out in your work, or out of interest if you want to carry out, you are feel free to interact with Dr. Thirugnanasambandam or to me. So we have the database with us, and any kind of collaborative publication or any kind of publication data sets we can give for more outputs.
Yes, sir.
Thank you.
Thank you. Thanks a lot.
Thank you, sir.
Thiru, thank you, Thiru. Thanks a lot.
Thank you, sir. Thank you, Lord.
I think Dr. Lata want to ask you some question. Virulenee, unmute Lata ma'am, please.
Sure, sir.
Yeah.
Sir, can you hear me?
Yes, ma'am.
Yes, sir. Really appreciate your work.
Thank
And
you,
also you have done the recent, as Dr. Inbakandan sir told about the recent databases. So it's a request to Inbakandan sir, if you can arrange on a program related this recent databases and give some basic explanation about it, sir.
Yes, ma'am. Yes, ma'am. Actually, if a particular group of people, because if we started narrating this database and bioinformatics based work to a common platform, it won't be very helpful. But we will make another workshop, and I will intimate to the WhatsApp group, so that those people who want to know more about this platform, particularly this genomic based or any kind of post genomic analysis bioinformatics work, we will arrange a workshop soon and we will inform you, ma'am.
Because he introduced some new terminology which is new to me. I don't know whether others may be familiar, like nano drop and TrueSeq kit, something like that. It is all new to me. So if you arrange any workshop like that, it will be very useful for us to explore more in bioinformatics.
Everything will be new normal. Even we will make an offline workshop also, and you are all welcome to our lab, and even the same instruments and other kind of work we will do offline also. But we will try, ma'am. We will try both offline as well as offline, online.
Thank you.
Thank you, ma'am.
Thank you, sir. Thank you, sir. Thank you.